Good morning. So today I am going to discuss an article from the International Journal of Nanomedicine with the title Graphene Oxide Copper Nanocomposites Suppress the Karyogenic Streptococcus mutans Biofilm Formation. So this was published in the year 2021 with an impact factor of 6.6. What is the novelty? So here they have the exploitation of antibacterial nanomaterials which can open up a new avenue for development of karyostatic approach. The aim of the study was to evaluate the antibacterial and antibiofilm properties of graphene oxide copper nanocomposites on karyogenic streptococcus mutants and the effect of GOCU, which is graphene oxide copper nanocomposites, on exopolysaccharides matrix synthesis and exopolysaccharide related gene expression were also detected, and the regulatory landscape of GOCU nanocomposites on S mutants pathogenicity was proved. Materials and methods. So what they had collected the materials, bacterial strains and culture conditions were analyzed. So the copper sulfate, how did they produce this copper nanocomposite? Copper sulfate solution was gradually added to prepared GO solution. GO is graphene oxide solution, and the final concentration of GOCU nanocomposite solution was defined as 1.5 mg per ml. And the S mutants wild type strain was obtained from the for the control group. The strains were grown in BHI medium, which is brain heart infusion medium, in an atmosphere of 80% nitrogen and 20% carbon dioxide at 37 degrees C. So these are the conditions for the growth of the Streptococcus mutant strains and how they produce the GOCU nanocomposites. And they determined the growth curve by using a 96 well plate and sealed the bacteria at 37 degrees C. The bacterial cell growth was monitored with a multi-scan spectrum, and the OD was 60 nanometers, which was measured at 1 hour intervals. So this is how they measure the growth curve determination. Colony forming unit counting method was used to count the colonies. And cell culture, live dead cell assay and cell counting assay was used, for which the human fibroblasts were seeded in 12 well plate at a density of 10 to the power 4 cells and cultured with different concentration of GOCU. And after culturing for 24 hours, the cells were washed with PBS and calcium propydium iodide working solution, and then they were incubated for 30 minutes in 37 degrees C, and the images were taken using fluorescence microscope. For live dead cell assay, they used the human fibroblasts were seeded in the same 96 well plate, and the CCK8 kit was used to access the proliferation of the human fibroblasts cultured with the GOCU. So what they did was they had plated the 96 well plates with the human fibroblast cells and checked the viability as well as the cell proliferation assay on the GOCU nanocomposite. And next they did the crystal violet microtiter assay, which was also used in 96 well plate. It inoculated the 2 microliters of exponential cultures of bacteria and 200 microliters of BHI, which is the culture medium, at different concentrations of GOCU. So they diluted the GOCU nanocomposites into various titers, and they checked which concentration is acting on the Streptococcus mutans bacteria. And they found the measured using the optical density, that is OD, with 575 nanometers. And confocal laser scanning microscopy was used, which is the biofilms were observed by using the argon and helium neon lasers. The green indicated the bacteria and red indicated the exopolysaccharide. In anthrone method, this method used 4 milliliters of anthrone reagent, which is added to the 1 ml sample solutions and heated at 95 degrees C for 6 minutes. And the absorbance of each sample at 625 nanometers was monitored on a microplate spectrophotometer. Then they studied the gene expression by qRT-PCR and RNA sequencing and data analysis. And finally, they did all the analysis using statistics SPSS software. The Shapiro-Wilk and Bartlett tests were used to assess whether the data were parametric or not. And for parametric testing, they used Fisher's test and one-way ANOVA to detect the significant effects of variables. For non-parametric testing, they used Kruskal values and least significant difference multiple comparisons were used. So the results were the antibacterial activity of GOCU nanocomposites on S mutants. The CFU decreased sharply from 10 microgram per group to 20 microgram per ml group. and they reduced the cytotoxicity of GOCU nanocomposites on fibroblasts. So it showed reduced cytotoxicity. The human fibroblast cells were viable at a concentration of 2.5, 5 and 10 microgram per ml of GOCU. And they characterized the biofilm, anti-biofilm effect of the GOCU nanocomposites, and they found that with 10 microgram per ml, it significantly reduced the compared to the control. And GOCU nanocomposites alter the S mutants biofilm architecture. So it not only reduced the biofilm formation, but it also altered the structure on whatever biofilm was formed. So GOCU nanocomposites treated biofilm showed small bacterial clusters sparsely distributed across the surface, and they impaired the exopolysaccharide production and distribution. So